My name is Jean Linsky. I'm the coordinator of the Global Conservation Consortium for Magnolia at the Atlanta Botanical Garden. And I'm very excited to be able to join you. Sorry I couldn't make it up in person, but very grateful to have this virtual option to share our work on pyramid magnolia. And yeah, it was great. I've been connected through Bob Askins and Dave Shrutram, so good to see you guys online. And thanks so much for, you'll hear a bit about some fieldwork they've joined us on and connected us with you all. So it's really great to be here. So yeah, we do have three speakers today. We're kind of going to do a tag team presentation. And so I'll let Martin and Michele introduce themselves before we get started.
Hi, everybody. Thank you for having us here today to talk to you about this project. I'm Michele Sanchez, and I work for Cooperative Extension at Clemson University since beginning of 2024.
I'm Martin Hamilton. I'm the executive director of the South Carolina Botanical Garden at Clemson University, and I've been in that role since the summer of 2023.
Great. Thanks. So yeah, you'll definitely be hearing more from Martin and Michele. And yeah, we will talk to you about this very exciting tree that I've come to love, the pyramid magnolia. Some of our work here in South Carolina and how it really connects to a global magnolia conservation initiative that I'm a part of at the Atlanta Botanical Garden. So we can go to the next slide. And so I'll first talk a little bit about why are we interested in trees? Besides them being very charismatic parts of the flora, we're starting to understand a lot more in the botanic garden and plant conservation field about the threats facing tree species. And so there's a big initiative called the Global Tree Assessment that's been going on for the last around 10, 15 years to really get a handle on the status of trees around the world. And one of the first steps was to understand how many trees there are. There's roughly 65,000 tree species in the world. And another main goal of the Global Tree Assessment was to understand how many of those are threatened. And so their goal is to create a conservation or an IUCN Red List assessment for all of those trees. And to date, what we found is around 38% of the world's trees are at risk of extinction. And that includes around 11 to 16. There's still some species we're sort of understanding the status of in the US. But yeah, around 11 to 16% of US tree species are threatened. And so that's from all kinds of different factors, over extraction, use in timber, habitat change. I think we all know what's facing tree species out there. And so, yeah, one of the big understandings within the botanic garden community is this idea that tree conservation has been going on sort of piecemeal here and there in a lot of projects. But because this is such a charismatic and threatened group, there's really a need to scale that up and to coordinate a little bit more between different groups that are doing tree conservation. And a lot of work that was happening actually was on similar species within shared genera that might have shared challenges. And so there's an opportunity to do a bit more sort of scaling up and synergy between organizations. And so Botanic Gardens Conservation International, which is a international botanic garden networking organization, came up with this initiative of these global conservation consortia, or GCCs, to really start to network people more together, use the specialist knowledge that people have, and to conserve trees where it was sort of needed the most. And so the Global Conservation Consortium for Magnolia, which I coordinate, was born out of this initiative. So we can go to the next slide. So yeah, as I mentioned, this is sort of nested within this Global Conservation Consortia initiative. So there's a number of GCCs that exist for other taxa. There's for oaks, cycad species, sort of quite charismatic but highly threatened groups. And so really it's a collaborative network, and we are global. We're going to talk about work obviously here in the Southeast US, but we do focus wherever there are magnolias that help. And so really it's about connecting experts, enthusiasts, and organizations, and developing and implementing sort of collaborative conservation worldwide. So next slide. So we, as I mentioned, we focus wherever magnolias are most needed or most need conservation. And so there are sort of six main hotspots of magnolia diversity that we work in, including Central America and the Caribbean, South America, and then East and Southeast Asia. But of course, Canada and the US, we don't forget about our 8 native magnolia species that are right here in our backyards in some cases. Next slide. So to do this magnolia work, we have a set of sort of objectives to think about integrated conservation. So all different aspects of what these species might need for conservation. And there's a few of them here. As I mentioned, really establishing and fostering a network of experts, so connecting people both within botanic gardens, but recognizing that there are many sectors that are interacting with magnolias and impacting their survival. We also do a lot to identify and prioritize which of the 360 some magnolia species around the world have the greatest conservation need and where we should focus our efforts. There's also a lot of applied research, so work such as genetics or taxonomy that can really help us support our conservation. So we connect with researchers in that way. And then, of course, increasing public awareness, getting the word out there about how great magnolias are, how much there is to be done. And in terms of sort of direct conservation activity, obviously ensuring that magnolias are persisting in the wild, but also what you're going to hear more about today is some of our ex situ or botanic garden outside of the wild collections, and ensuring that those have high conservation value. So next slide. So what we're talking about in terms of high conservation value is employing a concept that has really come up quite recently in the botanic garden world in creating not just separate collections that are at different botanic gardens, but really a meta collection, which is really just the combined holdings of a group of collections. And you can see here in my sort of schematic that I've put together, we're really thinking about for a given species, for example, there might be 4 gardens that hold that species separately at different locations. We might think about different gardens across the US, but really a meta collection is these gardens sharing both material and the information about their trees or their plants with each other so that as a whole, it has a lot of conservation value. We understand where the individual plants have come from. If one dies at a certain garden, we might be able to replace it or breed with another one if it comes to that. And so this really makes sure that all of the holdings at those gardens are really valuable for conservation. And these gardens are working together without one garden, for example, needing to hold hundreds and hundreds of trees, which it wouldn't have space for, but it can share across a number of different gardens. So you'll hear more about this concept and how we're putting it together for our focal species, which is the pyramid magnolia. So hopefully you're familiar, or if not, you're going to be by the end of the talk tonight. But this is the pyramid magnolia. As we all know, it's hard to get a picture of a tree in a forest. And this is quite an interesting specimen of the pyramid magnolia. This is really exciting to me, this photo, because it's actually the first pyramid magnolia I saw in the wild. So it was kind of exciting, although it's sort of a strange stature sort of over this ravine. But yeah, the pyramid magnolia is found across the Southeast US, from South Carolina across to Texas. I have a map in a few slides I'll show you. but it really grows on slopes and ravines in Music hardwood forests. Everywhere that I've seen it, it's on these north-facing slopes and these sort of ravines. Can get up to be about 40 meters tall. And really the pyramid name actually comes from, you can't see it in this picture, but it does create quite a nice pyramid-shaped sort of crown and habit. It looks quite nice when it's not being forced to slope over a ravine. And actually, in the next slide, if you go to, you can see some of the diagnostic features that really help us know that this is a pyramid magnolia. So you can really see those kite-shaped leaves, and with the really big ears at the sort of base of the leaf there. That's how I always know what it is when I'm walking through the forest looking for it. These really beautiful white flowers coming out in April, and then of course the pretty characteristic, bright red or pink cones, which is what we hunt for in April when we're looking for those the ripe fruits. So quite a stunning tree. I really come to love it. Um, and next slide. So as I mentioned, and as we know, it's quite rare in South Carolina, but pretty scattered actually across its range. And this map just showing the green states are where it's found. Those yellow and lighter green counties are occurrences, with the yellow being where it's quite rare, green a bit more abundant. And up actually in Kentucky, there's one county where we have a sort of little point, but that's actually thought to be extirpated, and we don't think it's there anymore. So yeah, quite a rare species across its range. And next slide. Um, so yeah, this is a bit about why we as a conservation consortium and network are, concerned and interested in learning more about the pyramid magnolia. Um, there was an assessment of the tree species of the US to understand vulnerability to climate change, and pyramidata, the pyramid magnolia was found to be relatively vulnerable in terms of habitat specificity and climate change. So it's actually assessed as endangered on the IUCN Red List. As I mentioned, it's really fragmented, and these, populations not being able to mix with each other across the range. And we've talked to several people and been out to look ourselves for various subpopulations that are actually declining in loss. And so we really wanted to understand as a conservation network, um, what's driving this loss? Are we actually seeing fewer populations? And what can we do? Where can we intervene to help conserve the species and make sure it can persist across its habitat? So next slide. So that was a bit of a background about it, and throughout the rest of this session, we're going to talk a little bit about some of the taxonomy and genetics research that helped us to really understand the species that we're working with. I'm going to talk a little bit more about our conservation gap analysis and some of the ex situ work that we've done. And then, yeah, we're also going to talk about the partnerships that have developed between Atlanta Botanical Garden and South Carolina Botanical Garden and others to help conserve the species. So I'm going to pass it over to Martin, who's going to talk a little bit about our taxonomic and genetic work.
Thanks, Jean. And as Jean said, one of the big things as a group, we're trying to understand what exactly is going on with this species, and that means that we've got to know what plant we're talking about. So over time, this species has been called its own species, a variety of another species, and so we've really got to know what populations of plants represent a distinct species so that we can conserve them and understand their status and develop management strategies. This also helps us to really think about areas of high biodiversity. So if cryptic species or a number of unique lineages are lumped all together, that doesn't give us an accurate picture of how highly biodiverse an area is. It also ensures that we're collecting genetic diversity from across the species range. And then, as Jean mentioned before, us being able to understand a species' vulnerability to climate change, land use change, and pyramid magnolia in particular, as she mentioned, from a climate perspective, but also what we're seeing from historic land use change, this species is very, very susceptible. So I'm going to start off with just a little bit kind of about the taxonomy. Um, so as I mentioned, pyramid magnolia has sometimes been treated as a variety of Magnolia fraseri. But modern work has really elevated it back to species level based on characters that we can observe in the field. And here you see from Weekly, Flora of the Southeast, they're treated as separate species, and what this boils down to is the size of stamens, the size of leaves, and the size of the fruits in particular. And then also, as Jean showed in the map, and I'll show another one shortly, they are separated as well in their habitat. And so Fraser magnolia grows in the mountains in the Piedmont, and the pyramid magnolia grows on the coastal plain. And here, just to give you an idea, these are fruits that we collected in the last couple of years from fraseri on the left and pyramid magnolia on the right, and you can see that the size difference, is nearly twice the size for fraseri as it is pyramid magnolia. And then, as I mentioned, once we start looking at those characters and we identify these as separate species, then the habitat preference becomes very obvious. So here the maps show very clearly coastal plain, from the fall line south for pyramid magnolia, and fraseri growing in the Piedmont, the edge of the mountains up into the mountains. And so for pyramid magnolia across its range, this is a screen grab from NatureServe. So each state, they do assessments on their species, and then those assessments get uploaded into the NatureServe website. And you can see at either end of the spectrum, pyramid magnolia in South Carolina and Texas has been assessed as critically imperiled. So it's a state level 1 species. And then it varies in between apparently secure to we have no status rank, to vulnerable in Florida and Georgia. And so one of the big things that we needed to do was ask questions about, um, do these two species really do show independently evolving lineages, or are they one evolutionary lineage? And to do that, we're going to use some genetic techniques to help us. And then the second main question is how well represented is the species in botanic gardens? So do we have good ex situ collections that represent the genetics that are found in the wild? And so to do that, sampling was undertaken. So lots of samples from pyramid magnolia, coming from herbarium specimens, wild collections, and then also from botanic gardens. And then a smaller sampling from Magnolia fraseri from herbaria and botanic gardens. I'll point out that there were 5 samples that the identification wasn't clear, but they were thought to be pyramid magnolia. We were able to have some really old material included in the sampling from a herbarium specimen from 1898. And then used 2 other magnolia species as outgroups. So Magnolia ashii, which is another southeast endemic, and Montebellonis was included as the other outgroup to help place the specimens. And the way that this work was undertaken was using 2 bait sets. So basically, a targeted set that is specific to Magnolia, which finds these 504 genes that are known and highlights those. And then Angiosperm 353, which is a universal bait set for angiosperms. And so between using those 2 bait sets, Around 850 genes were targeted from these samples. Um, this target capture method is really good because you can do both phylogenetic studies where you look at species relationships, but you can also do population genetic studies where you look at demographics and separation of populations of an individual species. Um, the other great thing about it is it allows us to include these herbarium specimens that often have very fragmented sections of DNA. And so we can look historically at missing genetics that we no longer find in the wild or we don't find in our ex situ collections. And so the next few slides, I'm just going to give you a snapshot of the findings. Don't worry about trying to read the graphs on the screen. The important thing is just the colors. And so on the left side of the graph is Magnolia fraseri. Those are blue samples. And on the right side of the screen are Magnolia pyramidata. Those are the orange samples. And it was very clear they formed two clades that are sister to each other. So this means that they are separate species, but are closely related. And then the five individuals that were unknown, those actually all fell into the Magnolia fraseri clade. So even though it was thought in the botanic gardens where they were held that they were probably pyramidata, the genetics were able to help us identify these as misidentifications, and they are Magnolia fraseri specimens. And the other great thing about this approach and having these samples from these botanic gardens is we could then understand how well represented Magnolia pyramidata is in botanic gardens. And so there was a paper published in 2020, which basically said that our ex situ collections are insufficient to capture the allelic diversity that's found in the wild. And so we need to do more work. We need to collect more samples from across the species range. It showed very clearly that certain lineages are underrepresented and others are overrepresented in ex situ collections. And so I'll just draw your eye over to the graph on the right. So those pink colors, the garden Magnolia pyramidata, you can see a lot of those cluster together. And what that means is they've come from lineages, from collections, from an area. And then there's big sections of that that are orange with no pink in it. And what that's telling us is out in the wild, there's lots of genetic diversity that we've not captured and we don't hold in our collections at botanic gardens. And so this really then highlighted the need for us to undertake a gap analysis. And that's the thing that Jean now is going to explain a little more about.
Right. Thanks, Martin. So yeah, we can jump right into the next slide. So when we talk about gap analysis, we might have heard about this in different contexts, but essentially what we're meaning is really understanding, sort of assessing the coverage and then gaps of both ex situ collections and in situ populations of target species. So really understanding where the conservation is needed and how we can address that. And so we put out a publication actually in 2022. You can see it here, the Conservation Gap Analysis of Native Magnolias of the US and Canada. You can find this online if you search for it, or I'd be happy to send people links. It's publicly available. But essentially, this is using a lot of, again, more methodology in terms of gathering data and analysis that is happening within the botanic garden world, but also used for a lot of other groups like crop wild relatives, so plants related, wild plants related to crop species, and is using a lot of spatial analysis that I'll take you through a little bit of what we did. So the next slide. Our gap analysis and that publication sort of assesses, as I mentioned, both in situ and ex situ status of various species. To put that together, we did both a literature review and surveys of people within the Botanic Garden and greater plant conservation community um, to get a sense of, so through a tree conservation questionnaire, so this was emailed out to a whole bunch of people to really get a sense of threats to wild populations for the species, what were the most urgent needs for those species, and to also get a sense of what are people already doing for conservation activities for those species. So it's a big thing for the conservation consortia to really get a grip of sort of what's going on, what are people working on, and how we can move forward as a network. So these tree conservation questionnaires are really helpful for sort of getting a handle on that. Um, so that was to get a sense of sort of those wild populations. What are the threats? What are the needs? But then we also did an ex situ survey. So this was sent out again to the Botanic Garden community with a lot of help from BGCI, so Botanic Gardens Conservation International. They have a huge network of connections. So we were able to do a species and accession level survey. So that was A, to find out which species are even in any botanic gardens, um, and also an accession level. So digging down a bit deeper to find out, how many individuals, if they're from the wild or if they're from more horticultural sources. And we were able to draw some of this information from Plant Search, which is a BGCI tool to gather information about what plants are in botanic gardens. But we also did a lot of direct emailing and questions for accession level data directly to botanic gardens. So next slide. We'll jump right to some of our results. These are pulled, this is pulled right from the report. And in this report, we obviously had the eight native species to the US and Canada, but we've tried to highlight where those results are for pyramidata. So when we were asking the Botanic Garden community, the plant conservation community, what the most significant threats to pyramidata are, you can see here, as we've already sort of talked about, these real concerns about disturbance regime modification. So thinking about fire and fire regimes that may have naturally been happening and how that's changing in the landscape for pyramidata. Obviously, development ongoing in the habitat, as we mentioned before, climate change and different land use practices. And the next slide. When we also then asked about what conservation activities was most urgently needed for pyramidata, what were these people thinking about? We had quite a lot of protection and management of habitat as a priority for working on pyramid magnolia, occurrence surveys and population monitoring. So we've sort of again seen this need to really understand what's going on with these populations. And then collection and distribution of germplasm. So making sure that that wild diversity is backed up in botanic gardens and sharing that with multiple botanic gardens. So we're already on the right path for what we're doing now. So next slide. So this is a result of that ex situ survey, so that dive into the botanic gardens to find out who and where pyramidata, the pyramid magnolia, is being conserved. We were able to even get down to the number of individuals that gardens were able to report to us. And pyramidata was actually one of the least representative of the US species in botanic gardens. Of course, macrophylla, tons of those in botanic gardens. But we can see that there's even less than 100 individuals of pyramidata. And the gardens were able to tell us the source or the provenance of their material. So we could get a sense of whether they knew if it was wild sourced. In some cases, gardens aren't sure where their material came from, or it may have been a horticultural source, so possibly a cultivar, or it had just been bred from sort of a botanic garden in many different generations. So yeah, we can see that there weren't a lot of botanic gardens holding pyramid magnolia. And so a lot of those gardens in the accession level, the information that they were able to provide to us was even if when it was wild provenance, the source, the exact location of where they collected that material from. And so we were able to put that onto a map, which you can see here. So those black triangles are the source locations of material that now exists in botanic gardens. And we were able to lay that over top of those lighter gray circles, the sort of estimated range, to really get an understanding. So that spatial sort of gap analysis of where the material was from and where it was missing. And so we can see right in that highlighted circle that as of 2022, when we did that gap analysis, we saw that there was no material from either Georgia or South Carolina in botanic gardens. And so that's a real gap for the sort of comprehensive collection and conservation of the species. So immediately we could identify some locations to go and scout to see if the trees were there, do some collecting. And so we were able to write a grant proposal to the American Public Gardens Association. And they have a fund with the U.S. Forest Service called the Tree Gene Conservation Partnership to help us get some funds to go out there and do that scouting and do that collecting. And so Michele is going to tell us a little bit about those trips.
Yeah, so I'm going to tell you a little bit of the survey we have done and how we developed the horticultural protocols for the species. So once we had the funding, then the team met regularly to discuss the project. We planned the field surveys and established collection targets. And we adopted the Atlanta Botanical Garden collecting protocols that you can see here. And they follow the Center for Plant Conservation, best plant conservation practices. And the CPC offers great materials, great guidelines, seed collection, genetics reintroduction for plant conservation. And this is a living document, so it's good to return and check what are the changes because it's something that is always updated. And they have several really good resources. And one of those is the CPC Rare Plant Academy. So if you're not so familiar with rare plant conservation or you want to refresh your knowledge, I really recommend this resource. And it's a course that, well, they have a course called Applied Plant Conservation course. And that course is really good and online and self-paced and is also free. So yeah, it's something that has come up in the past years and they keep adding modules to that. So how did we find these trees in the wild? So Jean talked a little bit about like how we target some areas. So what we did, we got the team together and started planning the fieldwork activities, the timings, and the locations. So we gather available distribution information from herbarium specimens, species accounts, and element occurrence data from natural heritage programs. And then we develop an ArcGIS field maps, offline map, that combined the occurrence records with the property boundaries and layers that showed us the topography, the land cover, and roads. And then these priority areas of high quality, suitable habitat were outlined, like you can see here in the middle. And that helped us a lot to target our surveys. So we are able to contact private landowners, often with the assistance from other organizations like Audubon Society, to get signed permission forms to access and to collect materials. And for the public lands, we acquired the necessary permits from state and federal agencies. And we ensured that access and permits were acquired in advance, and those records are really essential. So we used an Atlanta Botanical Garden developed survey 123 collection event survey form in ArcGIS to collect field data with centimeter accuracy coordinates using these externally connected GPS devices. And here I'm using the MLead or S3. In Atlanta, usually use the Bad Elf devices. But even in the forest and woodland areas, we got really good accuracy. And those data collected in the field were easily incorporated then into Atlanta Botanical Garden BRAM's database, and that we could export and then share with partners and with landowners. And we ensure that we don't disseminate any sensitive data. So when we do the reports, we do a public and a private copy. And the public copy is available, is published, and you can use this QR code here to download the report, but it's available on ResearchGate. The field work we did in 2024 was carried out in South Carolina and in Georgia. And once we had all the necessary permissions, we planned the field work to coincide with the pyramid magnolia flowering period, that's in April. And besides, it's a lot easier to spot them when they are in flower. It also gave us an idea of possible fruits that we could collect later in the fall. And in South Carolina, we surveyed public and private lands in Orangeburg and Calhoun counties. But we actually, in 24, we only found 26 wild trees in 2 private properties. And in Georgia, we recorded 59 trees in 3 different populations. And through this collaboration, we also could help build this capacity for the staff in both of botanic gardens, extension team, and also gave us the opportunity to observe this beautiful species in the wild and start planning some exit collections. In each survey location, we recorded high-resolution GPS positions, and we collected several different samples. Herbarium vouchers that were collected, you can see herbarium specimen here. They are now curated by Clemson University Herbarium. And DNA samples and shoot tips for tissue culture were also taken to Atlanta Botanical Garden. And on the far right here, you can see the little tips growing in agar. We collected seeds as well, so 107 seeds were collected in South Carolina from 2 trees only in 1 property. That's how we could find in 2024. And only we got 2 seeds in Georgia from 2 localities. And the seeds were all processed and stratified at Clemson and then grown at Atlanta Botanical Garden. And the aim of collecting all the seeds was to create a meta collection, like Eugene said, and develop these seed propagation protocols. So in 2025, we were able to secure another round of funding for continuation of the project, and we expanded the partnership to include Lady Bird Johnson Wildflower Center, who was gonna take the surveys in the western end of the species range that's in Texas. This QR code here in this slide is our final 2025 report that is also available in ResearchGate. And you guys can dive in there later. So in 2025, uh, surveys and collections for the pyramid Magnolia were done in Texas by Ladybird Johnson Wildflower Center, and that was led by Jonathan Flickinger, that you can see here in the middle picture. And the team was able to record like hundreds of pyramid magnolia trees. And the mature trees they encountered were reproductive, and the team collected also DNA samples, herbarium vouchers, shoot tips for tissue culture. And then they returned in the fall to collect seeds. Here in South Carolina, in April of 2025, we worked together with private landowners to expand the survey area of suitable habitat, and we actually found another 11 trees, what was fantastic. We always share the knowledge of the species with the landowners and land managers to help ensure that the habitat is preserved, and I was so nicely surprised that all of them were very helpful, very interested, and came in the field with us. And here on the left, you can see one of the landowners with this amazing pyramid magnolia that was nominated as the South Carolina champion tree for the species. And although we have surveyed like hundreds of acres of suitable habitat in South Carolina, we only were able to find 37 extant individuals so far, so not that many. But luckily, a lot of them were reproductive. We returned to those sites with reproductive trees in the fall, in August of 2025, to survey some more and to collect seeds. And we had a lot of help from landowners and also the botanical community, and we are joined by the South Carolina Native Plant Society, the W. Gordon Bess Arboretum, Moore Farms Botanical Garden, and Historic Columbia. And it was great to have everybody in the field. And this time we were a lot more successful, and we were able to collect 136 trees from now 9 maternal lines in South Carolina. And in Texas, they were able to collect 166 seeds from 10 maternal lines. So it was a lot more maternal lines that we were able to collect in 25 than 24. And we carried on also collecting some shoot tips for micropropagation. Now I'll tell a little bit how we processed these seeds and grew them to develop these propagation protocols for the species. So we started by collecting the fruits in the field when they are ripe. As you can see, this bright pink color. And we collect when the follicles are not completely open, but I think that's the best because then you don't lose many seeds. So what we do is we spread these seeds in trays or in sieves, and we always want to use fabric or paper bags. That's some breathable materials while you're transporting these fruits around. And then once they start drying, the follicles are gonna open and the seeds are gonna be exposed. So after we did that, we got the seeds out, and then we were able to soak the seeds in water for 24 hours, and then any seeds that floated were discarded. So after 24 hours soaking, then we remove the flesh, just rubbing on a sieve like I'm doing here. And these seeds are quite oily actually. So what we do as well is we wash them with a little bit of a dish soap and then rinse them. And then because these seeds are gonna be stratified for a while, so we sterilize them with 3% hydrogen peroxide to avoid the fungi contamination. So the sterilized seeds are ready now and then are ready to go in the vermiculite. So we use moist vermiculite in the Ziploc bags. And this stays in the fridge for a minimum of 3 months. It can stay longer if you need to. And so at this point, we passed the seeds back to Atlanta Botanical Garden for growing them. Atlanta sowed the seeds in their conservation nursery in January of 2025. And they used a customized media with 40% Pro Mix HPCC growing mix that has peat, chunk coir, perlite, mixed with 40% orchiata, that is aged pine bark, and 20% perlite. So quite free draining mix. And on top, they used sun grow propagation mix. And they used these deep flats and maintain the seeds in a greenhouse that is frost-free. The seeds then started germinating about 53 days, and we had quite a high success, about 94% success for germination. So once we had all this information, then we are able to put all that together into this seed propagation protocol. This way we can share, it's like a recipe on how to grow this species. So we can share that publicly. So this is published through our report and also available on ResearchGate. And we can share with partners, and when we are gonna give people seeds, we can send that. So it's a really good way of capturing all this information. So now I'll pass back to Jean, and she can tell you more about the education and outreach elements.
Thanks, Michele. So yeah, as we said a little bit earlier, one of our big priorities for the GCC for Magnolias is to share news and information about the work that we're doing. So on the next slide, we do a lot of communicating through social media. So we have a Facebook page, which I welcome everyone to join, and we have a LinkedIn page as well, and so we've connected with a number of sort of international species conservation initiatives. One called Reverse the Red, which is really championing species conservation, and so we've really committed to our work on Pyramid Magnolia, and we share that annually. They have a Reverse the Red Day in February, and so we post about this on our social media and providing updates and beautiful photos that we're able to take in the field. And so next slide. We've also shared a lot about this work in Atlanta Botanical Gardens publications. So we have one garden-wide called Plant Intel, and then Synecology, which is the Conservation and Research Department's annual sort of update newsletter. All of those are available online. You can definitely search those through ABG's website, and I can share a link as well. And so, yeah, that Safeguarding Magnolias in the lower left is our most recent edition, just sort of fresh off the presses and really exciting that we're able to share articles about this news with visitors that come to the garden, as well as people online who want to read about that. And we're really excited also that this news about this fieldwork has gone out in South Carolina Botanical Garden, their newsletter here, the Garden Gate. So we try and get that out to as many people as we can, and this is another opportunity for us to share. So we're glad to have been able to be here. So next slide. So what's next for our work with Pyramid Magnolia? So you can see here, we were able to sort of update our map of where we now have collections from. So we now have something in Botanic Gardens in in South Carolina and Georgia, and I was actually just packaging up some seeds today and sent them off to a few more botanic gardens. So it's really exciting that we've done that, but we can see sort of in southern Alabama there and Florida, and of course a few more sites in Georgia. There's more places to go and do some collecting. So next slide. So yeah, those are our next steps. So really getting all of those seeds that we've collected in 2024 and 2025. So the 2024 ones are now actually seedlings in our nursery, and we're going to grow them on for a little bit longer before we can send them out to a few botanic gardens. And then our 2025 seeds, we have, as I just said, I was just wrapping up shipping them off to different gardens around the US. And then we'll be, as a group, starting to build that meta collection and monitoring the survival and growth of those trees. We've got some more funding this year to do that scouting and collecting in southern Georgia and Alabama and Florida. And there'll be some more seed collecting from trees in South Carolina. And then as a group, we're doing more work around conservation planning. So thinking about how we can do more field work in different parts of the range, and going to check on different populations, and bringing new gardens on board into this network of people who are looking for and caring for Magnolia pyramidata. So next slide. So we just have to, I think we're wrapping it up, but I wanted to thank everybody who has been working on this project. It takes a lot of people in the field, in the lab, at the computer, to work on this project. So a big thank you to, yeah, our partners in the genetics research and at the Lady Bird Johnson Wildflower Center, and certainly to Martin and Michele for their leadership from the South Carolina Botanical Garden and Extension. And yeah, my team, we do a lot of work in the field at the Atlanta Botanical Garden. So it's as the story goes, it takes a whole network of people who are working on the conservation of this species. So thanks to our collaborators. And thank you to you as well for listening today. We've got our contact information up on the screen. I'm certainly happy to be in contact with anyone who has more questions about this species, and um, we yeah are happy to take any questions about our talk today. Thank you again so much for listening.
Does
I
anyone
have a
have any
question.
questions? Here we go.
Hey, Dave.
Yeah, my question is, with these populations being fragmented, then I think that each population has got different genetics, right?
Well, there there are differences in the genetics across the species range.
Right.
But when we get really close, so in South Carolina, we haven't done the fine scale work to know population level demographics at the state level yet. But there definitely is variation across the range, and populations or subpopulations do come out as distinct. But we that's why we've been collecting DNA samples while we're out in the field now, is so we're building up the required samples to be able to do a much deeper dive into the genetics.
Yeah. That's kind of like for instance, Eastern longleaf. There's differences in the populations, like from Virginia or from Florida. They're different. And I guess the next thing I'm going to ask, and then I'll let somebody else ask a question, is um, with these different genetics, then maybe it's a better idea to try to keep the ones that are growing, say, in Florida, with the botanical gardens in Florida, and same with South Carolina to South Carolina, instead of trying to mix things up, right?
Well, that depends. I mean, you can have situations where if you're allowing the plants from across the range to grow in association and cross, that you could wind up um, causing inbreeding or outbreeding depression. But the way that we approach these meta collections is when people sign up to receive material, they actually commit to not allowing the plants to cross in their collection and not collecting seed from those and distributing them. So we're trying to maintain those individual lines separated.
Okay, that answered my question because
Once,
that's where I was going to go with that. You hit on it.
yeah. Once we've done the deeper dive on the genetics, then you can do some work to actually do controlled crosses and try to help increase genetic diversity. But that's a stage much further down the road. Gene, do you want to add anything to that?
No, that covers it. Yeah, exactly. I think one of the big things we're also understanding is sort of where the plant can grow in botanic gardens. And so, yeah, Martin, you covered it in terms of being very conscientious about any crosses that happen or, having them not happen in collections. But we're also really interested in understanding where this plant can grow in botanic gardens and where it may not survive as well. So that's one sort of plus to certainly spreading it out a bit.
Sounds good. Yeah, I got a question following up on the DNA. And looking at the cladogram, it looked like there were 3 subgroups within it. Are there geographical links with those different groups, or is it sort of random, or you're not at a point where you can figure that out yet?
Well, I'll be completely honest. None of the three of us did the genetics work. We're presenting work that one of our colleagues at Atlanta did. But generally speaking, those areas that you see, the kind of breakouts, they do relate to distribution between South Carolina and Texas. And part of that can be driven by our sampling because, as I said, it's a good sampling size, but it is limited in that there were lots of areas that didn't have samples necessarily. So again, as we continue to work with this species and collect more material from the wild, and more botanic gardens actually share any new material that they've acquired, we'll be able to have a much better picture at a much finer scale.
Second question. Sure. You made reference, one of you, maybe more than one of you, made reference that climate change is a challenge. Are you thinking about that these locations are going to be warmer and drier, or is there something else about the climate that is a concern?
Well, I'll start and then let you guys jump in, but I think this also kind of relates to one of the questions online about changes in distribution. So, I mean, what we've anecdotally seen, and Gene mentioned this, is at least in South Carolina and Georgia, we only find the trees on these north-facing ravine slopes, and they seem to be at a very specific band above standing water, above rivers. We're not seeing the plants on south-facing slopes. We're not seeing much of any recruitment. And so I think historic land use change has really impacted where the species is now found. It's not tolerant to fire, and a lot of these areas around the ravines were cleared because it's a bit flatter. There's been pine plantations, there's been prescribed fires for forestry. And so I think we've wound up with these trees being limited in where they're able to persist. And then that's being compounded by, yes, drying, because we're having more infrequent rain, and then we get these really intense storms. Some of the trees that we were working with on one of the properties, there was a microburst that knocked down some really big oak trees and crushed five of these mature magnolias on one property. And when we're talking about we've got 37, 39 trees, that's a big percentage of them that were impacted by one single storm event. And so I think there's a number of pieces there that kind of relate to a changing climate. What Gene was saying also about sharing this material and looking at where its climate envelope kind of ends. So are there gardens further north that can successfully grow it, and give us an insight into, maybe its cold hardiness and whether or not it'll be able to migrate with our help or not, into new areas. But I think the big thing here is where we've got intact habitat left, we've really got to work with landowners, either public or private, and land managers if it's public, to ensure we don't lose that habitat, because these trees seem really, really tied to specific areas.
Questions?
I think you touched on my question, but my question is going to be about the seedling recruitment. Are there populations that have no seedling recruitment? And additionally, are there any populations that fail to produce seed?
Yeah, so we saw well, we didn't find many seedlings at all. And we found a small sapling that we couldn't relocate, so I'm not sure if it's still there. But one thing we think is that predation, so deer predation, has had a huge impact on the recruitment of the species. And we are not seeing, even though there are fruits, and there were fruits that have partly dispersed and everything, we are not seeing the seedlings coming back. So I think that's mainly due to this feral predation. And the other thing also is that in the first year, in 2024, that well, we had a lot less trees that we have located, so we didn't have that many to sample from. But also, we found fruits that had aborted a bit earlier and that were already on the ground, and that's why we couldn't collect from many trees. And so the idea is to investigate a little bit more with future grants, so what is happening with the recruitment, and try to see why the trees are not fully developing seeds. Even the fruits that we collected, some of them, like half of the fruit was empty. So we didn't get many fruits per, many seeds per fruit. Jean, would you like to add anything?
Yeah, I was just going to mention that it's sort of, it seems like recruitment is maybe a little bit variable. So some of the Georgia sites that we visited in 2024 did, in fact, there were seedlings that were maybe 10, 20 centimeters tall. I'd say we found quite a few of those. I haven't had a chance to go back since 2024 to find out if they've been, munched on by deer or if they're going strong. But exactly as Michele said, it's sort of one of our questions is what is driving this in different places, and the situations of the Georgia sites and the South Carolina sites seem very similar. So I have a big question about why these ones in Georgia were able to get this big, but we didn't see any seedlings in South Carolina. So some big questions for us and our ecology teams, and hopeful proposals in the works to be able to do maybe um, you know, um, some plantings where we might use exclosures to keep deer out, maybe some direct seeding and keeping squirrels out to sort of dig more into that question of what's happening to these seeds and seedlings.
But we do have direct evidence that deer predation in South Carolina is definitely a problem.
Mhm.
These trees are adapted to basal sprouting. So if they get damaged in the top, or if the canopy opens and they get light on the base, they'll send up basal sprouts. And every tree that we encountered that had basal sprouts, they had been munched on by deer.
Mhm.
And those trees that I mentioned that the white oaks had fallen on and knocked over, every branch that was within the reach of a deer had been nibbled. So they obviously go for this plant, and we talk a lot about this uh, in invasion biology, that we've got a lot of non-native species that the deer don't go after, and so they're selecting for those. And then there's a lot of native plants that they do go after, and so we're seeing a shift in the forest ecology where in a lot of these forests where we're working, there's hardly any understory at all. So the oak trees aren't having a chance to recruit, our magnolia trees are not having a chance to recruit, and you're winding up with kind of monoculture understories of things that deer don't like. They will eat them in certain situations, but it's stuff that they don't go after. And I think that's one of the things that's really impacting pyramid magnolia in South Carolina specifically, is that we've got very high populations of deer that are very hungry and they like magnolias.
Mhm.
Henry?
Are you seeing any disease pressures, either in situ or ex situ for this species?
For this species, we've encountered, I think, one tree that had some sort of canker on it. And again, I suspect that was from a really old tree fall damage that happened, which exacerbated that. But as far as pest disease problems, it's not been something that we've really noticed as an issue. And our colleagues in Texas, and the previous work that's been done on this species, nothing's really come to light to be of concern about, unless, Gene, you know of something.
No, no, it certainly hadn't. We hadn't seen anything, as you said, in the wild population. And I'm just thinking about contact I've had with gardens who are growing it, and I think um, in ex situ and botanic garden collections, disease and yeah, things like that hasn't come up yet. But something certainly we'll be keeping an eye on as we grow the meta collection.
And that's just another plug for, botanic gardens as well, because if we're growing these plants outside of their natural range, it can also be an early warning for us if a new pest arrives and it attacks them in cultivation, then we know this is a problem. So that's also one of the ways that we leverage the power of these ex situ collections is an early warning system for problems that may attack wild plants.
Any other questions? Sure.
I do. What pollinators? What are the pollinators for these magnolias? Any idea?
We need to find out.
Yeah, we don't know if genes know more, but usually magnolias are pollinated by beetles.
Oh,
Mhm. Yeah, we assume beetles as a sort of widespread magnolia pollinator. We actually have a PhD student from UGA who just got in contact with us, who's quite interested in pollination of magnolias and is going to be doing some work here here as in the garden on some of our specimens. And we have a few pyramid magnolias growing, so I'll certainly be asking her what she finds as she does her sampling.
But again, one of the things Michele mentioned before was a lot of the fruits didn't have any development like on one side. And that is quite worrying. We know that insect populations globally are crashing. And beetles in particular are susceptible to a lot of non-target pesticides. And so, that's one of the things we need to understand what it is that is pollinating the species. is it a suite of different beetles, or is it a very specific beetle that's doing it? And then what's its status? Because, our conservation of the magnolia may actually mean we need to do conservation of a beetle.
Hmm.
I would, this is not so much a question, but I'd just like to thank all of you for focusing the attention on this species here in South Carolina, for involving some of the folks here in South Carolina in the work that you're doing, and for things like developing the propagation protocols. I think that'll be tremendously helpful to us in the future. If I had a question, I guess it would be, what can we do? How can we help in the work that you're doing, particularly here in South Carolina?
I think for me, there's a couple of things that I would say. I mean, Bob, you and Dave have been very helpful in, one, trying to get us contact with landowners, but there's several properties that have historic records that we've not been able to get access to. A lot of that comes down to them being sold to LLCs and things like that, which tracking down the ownership is very challenging. But that would be one of the big things, is just getting the word out that, this tree grows on ravines. All the feeders into the Congaree River are target areas for us. If folks have land and have got decent habitat, we would love to get there and have a look. Joining in on those surveys, so having Native Plant Society come out and join on those surveys would be huge. There's lots of other associated species that grow in these areas that are also state priority species. And we've encountered a few of those during our surveys. So I think there's opportunities there, when we're looking for these plants, to find other things of interest as well. And then once we know where trees are, helping us monitor and collect from them. So the more hands, the merrier. You saw the photos. We had folks from Historic Columbia and Moore Farms came out and joined us, and it made a huge difference, just being able to cover an area really quickly, get the fruits, process the fruits. So anything like that would really be helpful. And we've had Upstate Chapter join us for some Stewardia collecting along the Chattooga River. It'd be great to do similar kind of things this year in the Congaree area with the Midlands, and we can make that known to you guys in advance when we've got plans for going out to collect.
All right,
And
just give us an assignment.
We had one more question in person, if y'all have another minute to answer that, and then we'll thank you for your time after that. Okay.
Yeah, number one, if y'all need help with collections or surveys, I like to hike and I'd be happy to do that. But my question was about planting in botanical gardens. Do you notice that is the plant able to grow in more exposed habitats after it's out of its seedling stage, or is it always shaded habitats, even for adult trees?
My experience is it varies. I know some of the gardens a little further north can get away with more exposure. But the work that we're doing and Atlanta's doing with the meta collections, we're purposefully trying to plant them in shaded habitat. I think again, it's one of the great things that we can do once we've got a good collection built up, is start pushing the envelope a little bit with them, and to see what their tolerance is like. Because again, and I guess maybe your question, if this is what you're wanting to know, there are lots of plants that their juvenile stages require pretty heavy shade for them to establish, and then they wait for a gap to form and then fill in that gap. And a lot of the trees that we've encountered, they appear to grow in mixed canopy and stay shaded most of the time. But there's been a few spots and a couple of trees that we've worked with that had a fair amount of exposure, particularly that property where we had the microburst. There were some large trees there that were literally right on the edge of the forest. So I think there's opportunities for us to research that a little more and try to determine, if we can get them to a certain stage and then do outplantings, can we get success with them if the habitat's been modified greatly?
Really, one quick
I
question.
would I would just add, I think that as we're building our meta collection network, we sort of plan to have these meetings where people can share exactly that sort of information, right, about where they're growing it and how it's doing. And we've had some of these meetings for another one of our focal species. So it's an exciting opportunity, I think, those meetings for people to share exactly that kind of information and how things are doing or properties in different conditions.
You had a quick question? Yeah.
Yeah, what's the closest relative?
Were y'all able to hear that?
Metric.
Did you ask what the closest relative was?
Yes, that was the question.
So from memory, like I said, I didn't do the genetic work, but from memory, Fraser eye comes out sister to pyramidata. But I don't know that we actually, Gene, do we have all of the Southeast US in a clade yet?
That is a very good question. I wouldn't be able to tell you right off the top of my head beyond that. Yeah, the closest relatives beyond the Fraser Magnolia.
So
Yeah.
conceivably, it can even be Asian. As a Asian.
He followed up with, could it be possible that the closest relative would be one of the Asian magnolias?
That's just wild speculation.
No, from a evolutionary perspective, that the Asian species and the U.S. species, they could potentially share a very deep history where there's a common progenitor for those, but they've been separated for so long. Our Southeast U.S. species will all be very closely related, and they will share an ancestor. And that ancestor may be the same as an ancestor shared with the Southeast Asian species, but it'll be very deep in time that ancestor would have occurred.
Thank you.
Awesome. It was wonderful to have y'all here. Appreciate the research you're doing and would definitely appreciate being involved further. I think everybody in the room would too. So
Excellent.